So I have the pleasure of introducing Mattias. He is um, he is attending Berkeley High School, and he worked with Dr. Rio um, Sanabria. Um, and Mattias, he loves dry mango, and he's really into backpacking. And his project is sensing and signaling ER stress by dopaminergic and serotonergic neurons.
Thank you, Israel. And so yeah, this summer I work with my mentor, Dr. Rio Sanabria, in the Dillon Lab, which generally studies aging and stress at a cellular level in the microscopic worm C. elegans. So we all experience stress on a daily basis, and many of us have different ways of dealing with this. That might mean watching TV or eating ice cream or hanging out with friends. Now, like us, our cells experience stress on a regular basis, and eukaryotic cells, which are compartmentalized into different organelles, have different stress responses for their different organelles. And I specifically studied the unfolded protein response of the endoplasmic reticulum, or UPR-ER. In the UPR, stress is able to be sensed by the ER, which uses the transcription factor XBP1S to turn on the UPR, which has two parts: chaperone induction and lipid depletion. In chaperone induction, misfolded proteins are fixed, and in lipid depletion, you're able to reduce the amount of lipids in the cell and also convert more of them into healthy lipids. Now, if you turn on the UPR in neurons, it's able to signal to non-neuronal cells to turn on the UPR and induces the chaperones and lipid depletion in these non-neuronal cells. And this has been shown in mice to increase insulin sensitivity and reduce fatty liver disease, and has also been shown to have other benefits. Intriguingly, if you turn on the UPR just in serotonin neurons, you're able to specifically turn on chaperone induction, but not lipid depletion. And by turning on the response in dopamine neurons, it only turns on the lipid depletion response and not chaperones. But the really interesting part of this serotonin dopamine signaling pathway is that both responses rely on this transcription factor, XBP1S. And my research is around differentiating what allows one transcription factor to elicit two totally different responses. Our first hypothesis is that serotonin and dopamine signaling use different transcription factors which work with XBP1S and are differentially recruited in order to elicit these different responses. And we would test this using CoIP, which is a method for pulling down XBP1S using antibodies, and also pulls down any other proteins attached to it, allowing us to characterize differences. Our other hypothesis is that serotonin and dopamine signaling change the chromatin structure, which means that they loosen the DNA winding around either the chaperone or lipid metabolism genes, allowing XBP1S to access them. And we would be able to test this using ATAC-seq and ChIP-seq, which use sequencing to characterize chromatin structure. And the significance of this project is it'll allow us to understand the UPR and its signaling better, which has many implications in fighting disease and aging research. Thank you.
Well done. And some beautiful slides there, Mattias. All right, so questions for Mattias?
I just have can you go over the chaperone thing, like how like what that is?
So yeah, chaperones are proteins that are able to go into the ER, and I'm not sure the exact mechanism, but these proteins are able to like refold proteins that have been misfolded and allow them to regain their function.
Okay.
Great.
I have a question. So how exactly can an understanding of UPRER be implicated in disease and aging research?
Okay, so when it comes to diseases, like it's been shown that the UPR has many different implications in different diseases, and also overexpression of the XBP1S and activation of UPR neurons extends lifespan in C. elegans. So if we're able to better understand how we can activate the different parts of it, you could use this to develop therapeutics, because it's been it's also can be negative to just like super overactivate the UPR because it can induce apoptosis and also sometimes lead to increased cancer. But maybe if we just induce the lipid depletion response, then that would only have beneficial effects without having similar negative pro-cancer effects.
I just have one more question. So do the neurons act like what? Do you know what certain neurons activate the UPRER? Or is it that the serotonin neurons and the dopamine neurons, like they both, like certain neurons, like I don't know what I'm trying to say, but like, is it like specific, or are there just types of neurons that activate the UPRER and then activate both the lipid and the chaperone stuff?
So most of this research is based on C. elegans, where serotonin and dopamine neurons are like very specific. And it's been shown that it's through like neurotransmitter release. So we know that if you knock down serotonin or dopamine production, you block these two chaperone and lipid depletion responses, and that like small clear vesicles, which are used to release neurotransmitters, are required in order to turn on like the normal UPR just through pan-neuronal activation. But the exact pathways aren't completely known.
Okay, thank you.